Thank you very much, Rachel. Actually, it is a great honor to be here. As Rachel said, I grew up in Leeds. I lived here until I was 18, and I kind of went off for various places, including Durham and other places, came back here. So it's a bit of a homecoming. And indeed, in the, I guess it was probably the late 80s, I remember my parents used to come to the Phil and Lit. I didn't even know it was called the Phil and Lit, but looking back, it clearly was that. And probably I wasn't at all interested in it at the time. But so, yeah, it's an honor to be here. So, as Rachel says, my career has been quite eclectic. I'm now head of the School of Computing. As you can see, we're about to change our name. We're going to be Computer Science. You can maybe ask me at the end what I think the difference is. But I'm a physicist by training, and I've kind of had a career that's kind of jumped between different disciplines. But a common theme throughout it has been optics and light and instrumentation behind light and optics. So I'm going to kind of, I hope I'm not going to jump around too much today, but I'm going to take you through some of the things that I've done in my career, which hopefully will be of interest. So I'm going to start with my conclusions. Click on that. I'm going to start with my conclusions, which many of you who are scientists, I hope you kind of agree with, and it's that advances in science are often very much intertwined with advances in instrumentation and techniques and how to do stuff. And you know, I've kind of the things I'm going to talk about today: telescopes, microscopes, and the physics of the eye, or the optics of the eye. I mean, telescopes and microscopes were invented hundreds of years ago, but they're still very much an active area of research. And as we develop techniques in those, they lead to further scientific understanding. You know, you might think that, you know, a telescope's a bit kind of old hat. It's been around for a few hundred years, but I always think back to, you know, when Galileo or whoever it was first kind of looked through telescope. I mean, what a revolutionary thing it was to be able to see things in the sky that one could not see with your naked eye. And there's similar arguments when we kind of looked down and could see things down a microscope that you couldn't see with a naked eye. This was a revolutionary kind of idea. And telescopes and microscopes, of which there are thousands of varieties, have taught us much that we know about the world. My second conclusion.
Yes. I don't want. Well, you can see that. You can see what's going on.
Second conclusion, which again I hope is not a surprise, is that science and technology doesn't follow a linear development. I'm going to jump around between different subjects a little bit, and it kind of shows the way you work in one area and it can kind of lead you into other areas which don't necessarily seem connected. So I'm afraid when I want to advance the slides, you have to click on the yeah. Okay, so I'm going to tell you about a technique that I've and others have developed over many years, and it's called adaptive optics. And this is the, if you like, the theme throughout the lecture. And I'm going to show you how it's been applied to astronomy, microscopy, human eyes, and also what I hope is a bit of a fun project at the end that I did on animal eyes. It's good double act we're doing. No, but the problem is the mouse needs to be clicked on the screen. It doesn't advance. Okay, just to remind you, okay, the electromagnetic spectrum, or what is light, extends all the way from, you know, radio waves, which are meter wavelength waves. You're probably all old enough, you remember that the radio is just a broadcast there. Their wavelength 275, 285 is engraved in my head from my childhood. I listened to Radio 1. So then, yeah, the optical spectrum, the stuff that we see is only a very narrow band of light in the middle of the spectrum, and then we go the other way. We have X-rays and gamma rays, and most of this stuff tends to be blocked by the atmosphere, otherwise we get frazzled. But the sun is giving out its peak peak wavelengths in the visible spectrum. And I guess there's no coincidence that matches where our eyes are sensitive. So just to remind you, this is background where we're talking about. And then also a reminder, there's a couple of pictures I've grabbed from the NASA website. They're taken with the Hubble Space Telescope. And they're two pictures of the same thing. It's called a Lagoon Nebula. And the reason I've shown these pictures is to show you that when we look at different wavelengths or different parts of the spectrum, we see different things. So these are both the same object, but we're just looking at different wavelengths. So the top is at the visible light, and we very much see structure formed by gas and dust. And then down at infrared light wavelengths, which pass through that gas and dust, we see a completely different structure. So that's by way of means of background. Let me ask a rather strange question, because it's kind of not very well defined, but I think it's quite a fun question to ask. What's the best we can see? It's kind of not a good scientific question because I haven't really defined what best is, but I'll ask it in a loose sense. I mean, generally speaking, when we're interested in either seeing with our eyes or seeing with an instrument, we're interested in either fine detail, that's probably the most important thing, or things which are very faint. So what ultimately governs how well either we can see ourselves, or how well can a telescope see, or how well can a microscope see? It's what is known as one of the fundamentals of physics. It's called diffraction. And the rather annoying idea, which is like one of the basic principles of physics, annoying because it makes all our instruments worse, is that if you have a wave, this could be waves on the sea or a wave of light, as they pass through an aperture, which you're seeing on the top left, they kind of spread out. And very simply, the narrower the aperture, the more they spread out. This is one of the basic pillars of modern physics, and you may have heard of the uncertainty principle, but that is it in a nutshell. The narrower the object, the more things spread out. So in general, it's good to have big apertures in science. And I'm not going to have, well, I've got one maths equation. I hope you don't mind me showing it. The angle, I'm looking down at the bottom of the slide, the angle that the light spreads out by goes as the wavelength of light divided by the diameter. So if you want to make fine details, fine images of objects, you want to have that angle as small as possible. So this equation is telling you want to go to short wavelengths and big apertures. So that's fundamental. Well, telescopes are big for two reasons. One, they collect more light, the bigger they are. And secondly, equally importantly, they can see in finer detail, the bigger they are. So I just wrote some numbers down. Here's some calculation. Let's see how fine we can see. So a 5 millimeter pupil size in your eye would be like that's when your eyes are dilated, about as big as you can see. If we do that calculation, we can see around about 1/100th of a degree. This is if you've got tip-top eyes, so you're 20 years old and you don't have any vision problem. You can see about 1/100th of a degree. How does that relate to a distance? Let's imagine you're looking at something 25 centimeters away. That's about as close as we can focus. That's 0.03 millimeters. Probably you can guess, yeah, I could probably just about see that. Or far away, that's how far, if you look at the moon, that translates to 70 kilometers on the moon. So that's the finest detail you can see. Microscopes, on the other hand, the smallest things we can see, that's a kind of a bit simpler, really. So is the wavelength of light. So with a microscope, we can see things in visible light down to about 0.5 micron, 0.5 millionth of a meter. With telescopes, I did a quick calculation about the world's largest telescopes around, well, visible telescopes around about 8 meters. We can see down to probably around about 5 kilometers on the moon. That's the best we can see. Or in theory, that's about the best we can see. what's the highest resolution that's ever been taken by a telescope? Interestingly, that is not taken with a visible telescope. It's taken with a radio telescope. And with a radio telescope, it's basically a lot easier to kind of do the instrumentation at radio wavelengths. So that even though I've said here it's good to have short wavelengths and radio wavelengths are much longer, we can synthesize telescopes by taking a telescope in the UK and one in America and combine the light together, and we can synthesize a radio telescope that's basically the size of the world. So we can have a huge, huge aperture. So that gives you some kind of idea as to the scale of things. But bear in mind, this is theory. This is the best that physics can deliver to us, okay? In practice, you know, we have microscopes, we have lots of issues with imaging through things. If we're having you and I, we have various stuff going on in our eye. We're all just about wearing glasses. And if you are a telescope, you have to kind of look up through the atmosphere. And the atmosphere is a big problem. It's conveniently, whoops, what's gone here? The atmosphere conveniently keeps us alive and gives us oxygen and allows us to live. But if you're an astronomer, you've got to image up through this horrible layer of stuff which completely distorts the images. So this was a video which I don't. Amazing, it worked. We did a few issues earlier. Right. This is a real picture of an astronomical observatory. This is on the Canary Islands, the islands of La Palma. It's one of the less visited islands. I recommend you go if you can. It's beautiful. This is the UK's what's called the William Herschel Telescope. This is real. This is not photoshopped on. This big green line. It's a laser that's been shot out of the telescope. And if I go to the top, what we're seeing there is simply an image of a star taken through a telescope at fairly high resolution. And what you would expect to see is a little pinprick of light, I assume. But when we magnify it up, you see this big kind of boiling pot of horribleness from the point of view of an optical person that's spread all around. Now, what you have to imagine, if I kind of stop it there, if there was no atmosphere, all the light would be concentrated into kind of tiny little spot, maybe that size there, and it's spread into this huge area. This really corrupts the images that are produced by telescopes. I mean, they're really kind of awful compared to what they should be. And this is one of the reasons why we want to put telescopes in space. We have the Hubble Space Telescope, or there's now the James Webb Space Telescope. We get around this problem. But putting telescopes in space is very, 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 very expensive. And you still can't put a really, really big telescope in space for obvious kind of practical reasons. So adaptive optics, which is the theme of my lecture today, is all about trying to produce images on the ground that we could have taken in space. So here's a very old image, actually, but it's of a binary star. So two stars which are next to each other. And if we just look through the atmosphere, and normally we have these images blurred into the one. And if we have an adaptive optics system, we can separate the two out. So I'm not going to spend hours telling you how all this works, but I'll tell you the basic building blocks because it's kind of relatively simple. So this is a schematic of how the system would work. So this is meant to represent waves coming into my telescope. So this is in the case there's no atmosphere and everything's perfect, and we produce a nice sharp image. With the atmosphere, light comes in. Initially, it's kind of good quality light. It's traveled for literally billions of years perfectly through the vacuum of space, and then in the final microsecond, it gets messed up by the atmosphere. So this is what we want to fix. And I kind of got this block diagram of how an adaptive optics system works. It's one of those things that's kind of a huge amount of devil in the detail, but schematically, it's fairly simple. We have light. This is my telescope up here. We have light coming in, and we bounce the light from a mirror. And it's literally a mirror whose shape we can deform and bend. And if we can make the shape of this mirror be effectively the opposite of what the atmosphere was doing, when light bounces off it, we get nice corrected wavefronts. So then, of course, we need to know what on earth do we make the shape of this mirror. So we have to siphon off some of the light and we send it to what's known as a wavefront sensor. This is a device that tells us what the atmosphere is doing. So we have them connected into a feedback loop. And this kind of feedback loop is a kind of a classic thing in engineering. It's okay, same kind of principle as how your central heating works in your house. You have radiators, and which is a correction system, and you have something you're trying to correct, it's temperature, and you have some kind of measurement system, which is normally a little thermometer in your wall. So control engineering is really kind of fundamental process. Right. Oh, no, don't do it.
No.
Don't end meeting.
No.
That one.
That one. Thank you.
Can you share your screen, please? That's disappeared.
Ah, thank you for that. Why is the screen? What have we done? Eric, help us with stress. You'll be pleased to see head of computing has equal problems with technology.
Have to,
go to all of the here.
There, we lost the share screen.
we come out of Zoom.
We come out of Zoom.
Oh.
Can someone online speak? Can you still hear me?
Yeah, I can hear you loud and clear. Yeah.
Clear.
But you can't.
And it's great as well. Really enjoying it, Gordon.
Oh, thank you. But it's correct. You can't see any slides still.
No,
Only on your screen.
We've only got the view of the room at the moment.
Of the room.
So
Okay. Well,
if it's not very clear on the screen that you've got in the room, we can't see it very clearly. So we haven't got a shared screen anymore.
But you're painting pictures in my imagination, Gordon.
That's your PowerPoint.
So we can see that on where you are.
Yeah.
But we haven't got it separately on our own computer screens.
It's like we're actually in the room at the lecture, though, isn't it? That's what we'd be seeing if we were in there.
I think we're seeing it as coming through from the recording mode.
Yeah, no, I understand. Yeah.
Thank you.
Thank you.
Run out and get a little kid. They'll know what to do. Head of computing. Huh.
If you click on the little black screen and make it full screen.
I think you've minimised that green arrow in the bottom.
Okay.
There you go, now share.
Ah,
There you go.
yeah. No.
Yeah.
Share.
Oh, it's that one there. isn't it?
I think we're back.
Can everybody see the diagram?
Yay!
That's it.
Yeah,
Love it
I can
when
see
when
it.
a plan comes together, as a great man once said.
Could everybody now mute themselves?
Yes,
please?
that's good.
Sure, this is a ploy to humiliate the new computing. Great. So adaptive optics, a technique to look through the atmosphere. And here's a fantastic image taken by the European Southern Observatory of Neptune. Hopefully obvious to you that the one on the left much better. So we're kind of getting resolutions from the ground that we could get in space. So how to build bigger and better telescopes? The next, our plans in astronomy at the moment to build well, what was originally going to be a 100-meter class telescope, 100 meters, let me let that sink in. That's a telescope that's 100 meters across. I don't think we're quite as ambitious anymore, but there are still plans of various telescopes around the world, 20, 30, and 40-meter class telescopes, which are going to have absolutely fantastic resolution. And although I've explained adaptive optics rather quickly to you, they're going to have them fitted, and are going to be absolutely integral to the operation of the equipment. Okay, let me move on briefly now to talk about applying adaptive optics in microscopy. I'm going to be very quick on this because it's essentially the same problem. As we look down a microscope, often in a microscope, you prepare samples and you slice them up and have them there as a nice 2D image. That's relatively easy. The problem is when you want to image through thick specimens or even more image in vivo while something's living. And so this whole technique of adaptive optics has equally well applied in microscopy. And I think in some ways, there's kind of some real big challenges there. If you just want to image through a thin layer of something, adaptive optics is helpful and important. If you really want to start imaging through thick layers of stuff, then, well, that's work in progress. We're kind of working on that, and progress is being made, but there's a technique based around what's known as ballistic photons. So if I send a beam of light in my hand, basically it doesn't go through it, but there's literally millions of photons and maybe 1 in a million goes through. And if we can collect that light and analyze it, maybe we can start to image through things. Obviously, X-rays are what people use to look through people, but X-rays aren't very nice, and it'd be much nicer to use light. So here's an image I took with my team in Durham of this is essentially a slice of a heart of a zebrafish embryo. And so this is about 100 microns, tenth of a millimeter in size. And what was really interesting about this image is this was taken in a real live zebrafish. Okay, so its heart's literally beating, and we had to image through the rest of the zebrafish and then sort of build up this image in real time without killing the zebrafish. So adaptive optics was key to that. Let me move on now to talking about the human eye. This is more work that I've done more recently. So let me ask the question, where else are adaptive optics used? I mean, I've talked about big telescopes, which cost literally millions of pounds or hundreds of millions of pounds. And I've talked about expensive microscopes, which are used in labs, which again are very expensive. What about kind of more everyday use of the technology? Well, the first thing is that your eye clearly has some kind of adaptive optics system in it, because looking around, there's probably 1.5 people in this room who have the ability to focus. So I'm making assumptions of people's age, okay? So we can all focus when we're young. When we get to about the age of 45, we have lost that ability. But nevertheless, our eyes can focus, and there clearly needs to be a correction element. There's a lens in your eye, you probably all know that, and it changes thickness, which enables you to focus. And then there's some kind of feedback mechanism that which tells it whether or not it's in focus. So I'm going to come to that in a minute. Interestingly, CD players, I guess CD players are kind of becoming old technology now, aren't they? But CD players have a system, because there's a laser which reads out the disc, and it needs to track the focus, and it needs to make sure the laser's in the right position. There is a little adaptive optics system which probably costs $10 to make in a CD player. And then a really interesting area, switchable spectacles. So this picture here is a company called AdLens, and it was interested in making spectacles for the developing world, where there's a huge number of people around the world who don't have access to eye care. And if you can't see properly, it massively limits your chances in life, because if you can't read, many people can't get a job, etc. So there's a whole slew of technology that's out there, but I thought these were fascinating. In fact, I used to have a pair. pair of spectacles with syringes on them. It's very simple, and you can pump the syringes in and out, and then tune your spectacles. And the idea is you don't have to walk around with syringes on your head. Once the focus has been set, you can kind of keep them fixed and remove them. Or indeed, again, a lot of work in development. How do we have spectacles which switch? I guess I'm wearing bifocals, and probably many of you are as well. But in some ways, it'd be nice. I'm kind of amazed that bifocals work in a way. Having a lens with two different areas in them, it'd be nice to be able to switch the focal length of light. So big area of research there. Let me move back to the first topic I was talking about, the eye, because I think this is kind of an interesting basic science question. You think we would know the answer to this question, but it's not yet solved, and one of the things I'm working on. So how does the eye know how to focus? We know there's a lens in your eye, and we know the lens can change. There's muscles around it, but how do we know when the light's in focus? And, you know, imagine I look at something close to me and then I look at you a different distance away. How do I know which direction I need to focus? How do I know whether to make my eye more powerful or less powerful? So that's important because it's a basic question in science, but I think it's also important from a practical point of view. If you want to build a virtual reality display where focus becomes important, you better understand how the eye works. Otherwise, you're not going to be able to replicate it properly. Or indeed, if you wanted to build some kind of prosthetic eye, if we don't know how the eye focuses, how are you ever going to build a perfect prosthetic eye? So it's an interesting problem, because this is a simulation I've done of this is just a test pan. Doesn't really matter what it is. It's just something with lots of detail in it. And on the top, this top diagram of things are nicely in focus. So we measure things in the eyes in these bizarre units of diopters, which is an inverse focal length. I'm sure you're familiar with them from the opticians. So this is perfect. What's interesting is if we defocus in either direction, so we either make the lens stronger or weaker, it has the same effect. So plus 0.5 diopters looks the same as minus 0.5 diopters. So how does your eye differentiate between these two? We know from experiments your eye can do it. So how does it do this? So I won't go into all the ins and outs, but I'll tell you what the three main ideas are. I won't do them in order. I'll come to what I think is the best one last. So the first idea is we use what I know as higher order aberrations. So the most familiar of those is astigmatism. I'm sure many of you have got glasses which fix astigmatism. You take your glasses off and rotate them, if the light kind of bends all around, you've got astigmatism. So in astigmatism, basically, if you look at an image out of focus, you see horizontal lines sharp and vertical lines defocused, or vice versa for the other side. So in principle, we could use these. I'm not convinced that's a very good cue, but in principle, we could use those. We could use, what we find is that the eye, when it's focusing, it's constantly in motion all the time. It's vibrating all the time. It has micro fluctuations of accommodation. Accommodation is the other word for focus. So it could be using this as a probe signal. I don't think that's very likely, but there's literally hundreds of papers written on this. I actually think that the best idea, my views on this change every few years, but anyway, my current thinking of the best idea is that we're using chromatic aberration. So your eye is quite a bad optical instrument, and different wavelengths have different focus at different distances. So blue light has focused at one point, green light at a different point, and red light at another point. And frankly, it's amazing that when we look at the world, we don't see colored fringes all over the place. Somehow our brains adapt to this. But we could use chromatic aberration as a cue to how our eye focuses. And in fact, I think there is very good evidence that we use that. So really, really interesting question, I think, and a question that's kind of still very active and still very open. And as I said, really important, both practically and from a basic science question. Okay, just some pictures showing what I just said. So this is a picture of how astigmatism could be used as a cue. So top left, if we have a perfect image, if we have the best we can get, if we've got astigmatic eyes, we'd see something like that. Whereas if we defocus either side, you can see you start to get either vertical sharp contours or horizontal sharp contours. So we could use this as a cue. This is an interesting photo. It's again slight diversion before I move on to animal eyes. I'm sure many of you might not have seen this particular picture before, but you'll probably be familiar with the effect. What is this a picture of? It's a city, yeah.
Skyscrapers.
You don't have to. I'm not asking where the city is, but what's special about it? What does it look like? It doesn't look like a regular city. Model. It looks like a model city. It looks tiny. It looks miniature. And this is what's called a tilt shift effect in photography. So this is indeed a real picture of a real city, and we've artificially blurred the image top and bottom. And if I show the original picture, you just see it's a completely normal image. So the reason I'm showing you this image is that, we think of, certainly in terms of scientific instruments, that things which are out of focus are a bad thing and just a problem that we kind of want to get rid of. But actually, the way blur or defocus affects an image can actually affect our perception. And this is a great example of that. I mean, once you see it, I think the effect is very powerful. You convince yourself that that is a model. It's hard to convince yourself that it isn't. And of course, what's going on is that when we look at models, when we look at things which are close to us, we're used to having a big change in defocus. The closer we bring something towards us, the more the focus changes. So your brain is looking at this and saying, oh, it's sharp in the middle, defocus close to me and far away. That must be a model. So how we see defocus affects our perception of the world. Let me move on to my final topic now, which is about animal eyes, which kind of seems a long way from adaptive optics, but it's kind of my key point, that kind of science sometimes takes tortuous routes and kind of get involved in different things. So this was the question we tried to answer. Why do some animals have elongated pupils? This is Durham, by the way, so an old slide I copied over. So here is a sheep, and you see they have these very distinctive horizontal elongated pupils. So why is the question? And we also get animals which have vertical pupils, cat being the most obvious animal. So we get animals which have round pupils, and we get animals which have vertical oriented pupils, and we get animals with horizontally oriented pupils. We never get animals with 45 degrees. There's never, there is vertical, horizontal, or circular, never some random angle in between. There's a fox. Interestingly, foxes have vertically oriented pupils. Dogs don't, foxes do. Snakes and lizards, very famous for their slit-like pupils. Again, they're vertical. Interestingly, a tiger has circular. Tiger is clearly related to a cat, but they've got circular pupils. There's an old picture of me. I'm a human. I've got nice round pupils. Horses. It's actually quite hard to see the pupil of a horse because horses tend to have dark irises, but you can get horses that have these light-colored irises, and you can see they've got horizontal pupils. Moose, and as I already mentioned, sheep and goats, horizontal pupils. So, if we kind of try and do the taxonomy of this, we've got vertical pupils. I mean, we're being scientific. Sometimes we have animals subcircular, like they're almost circular, circular and horizontal. And then we looked at that in the way animals get their food. And we can quantify them as prey animals, animals that get eaten. And then prey animals, animals that eat other animals. We can further divide those into what are called active predators or ambush predators. So an active predator will be like a dog. It kind of runs around and chases down its prey. Or an ambush predator will be like a cat. A cat sits really still and then pounces on its prey. And then we also had to look at when these animals are active, day, night, or polyphasic means day and night. And we did a lot of analysis of this. And so each of these data points that you hear represents a different animal. And you can see we kind of, the individual points tell me the time of day they operate. I've got the shape on the y-axis and the how they collect their food on the x-axis. And you see the points aren't randomly distributed. There's a nice correlation. So in general, if you've got a horizontal pupil, you tend to be an animal that's eaten by other animals. And if you're up in the top right-hand corner, if you have vertical pupils, you tend to be an ambush predator. So that's it in a nutshell. So what does this mean? This says the same thing. Species with vertical pupils are ambush predators, and they tend to have eyes facing forward, and they tend to have stereopsis, is the ability to see in 3D, something we've got. Horizontal apertures tend to be in animals which are prey. They tend to be grazers. They tend to be eaten by other animals, and they have lateral place eyes on the side of their head, and they have no ability to see in 3D. So, okay, I won't go through all the arguments. There's a sort of a lot of literature on this, some of which we kind of showed which was wrong. We came up with in science, it's really good if you do you want to work out a correlation, but a really kind of killer point when you're doing science is if you can make a prediction of something new, because that shows there's something kind of valuable in your theory. And we predicted that actually this effect would be more prevalent in animals which are not tall, animals which are close to the ground. And indeed, we did find that. We found that animals which are relatively smaller have a greater preponderance to having slit-like pupils. So, for example, a cat has a vertical slit, where a tiger, which is taller, doesn't. Let me jump past this. So, why is this? I won't go through all the arguments in the ins and outs, but this is it in a nutshell. An ambush predator, if you're a cat and you need to pounce on a mouse, you need to accurately measure the distance to the mouse. And you can't do things like judge the distance by moving your head around, which we might do because that gives away where you are. So you can only use depth from blur and depth from stereoptics to 3D. And it turns out that having vertical pupils aids you in making those judgments. Let me get past this. The horizontal pupil is a really interesting argument. It's interesting because what we showed that is if you have a horizontal pupil, you basically can see in a bigger kind of range of views. So if you're out in the savannah in Africa and you're about to be eaten by a lion, it's good to have a horizontal pupil because you can basically see wider. Now, the reason that is an interesting argument is that if you're not kind of doing optics on a daily basis, you might think, yeah, well done, Gordon, that's fairly obvious. I can, you don't need to be a scientist to work that one out. But actually you do, because if those of you who do photography know that if you change your pupil in your camera, it doesn't change your field of view. So actually it's a non-trivial argument. The fact that we have horizontal pupil gives us a wider field of view. Is down to the intricacies of the optics of the eye. So it's, I like it because it's a kind of really obvious result, but actually it's not all obvious if you think about it. So wider field of view from horizontal pupils. That gives you some of the calculations that we did. Now then we realized there's a really interesting thing that I think we were the first people to observe. You can go observe this yourself. It's dead easy once you've seen it. If you have a horizontal, so I'll stand here so people online. I'm a sheep now, okay? So I've got horizontally oriented pupils, okay? So these are my fingers and my pupils. And there is a problem that when I decide I'm going to eat, I bend down and my pupils are no longer horizontal, they become vertical. And maybe that's going to mean that I'm more likely to be eaten because then I can't see behind me as well. Same is true in horses. And what we noted is that as the animal bends down, I hope you can see it in this picture, the eye is essentially rotated to maintain the long axis parallel with the ground. Okay, so as I rotate, these are my pupils. As I rotate, my eyes are kind of doing this. If I project that round onto my eyes as a human, that's like it's like an ability to kind of turn your eyes through an axis that's kind of like that. We can actually do that, but only a tiny little bit. But we've observed that that these animals have this. It's called cyclovergence ability. I don't think this will work. True. Oh, it does. So what you want, I'll play that again. So look at the sheep's eyes. All the time they remain, the pupil remains horizontal to the ground. You would not believe how long it took me to take that video. I have spent more of my life, living in Yorkshire, it's no problem finding sheep to video, but God, are they difficult creatures to video. I even had ethical approval from the university ethics panel because I was like, hang on, is this research on animals? Do I need to just get approval to go around with my camera? Likewise, this picture, this was like, you can see that this horse was selected because it's got this nice blue pupil. Blue irises, so we can see the pupil. We basically went on the internet and said, hey there, well, has anyone got a horse? And there was a kind lady in Teesside who said, yeah, I've got one, you can come on video. But yeah, you can imagine me going into the university ethics panel. Oh yes, I found this woman online who got a horse and is just saying. So yeah, cyclovergence and sheep. This is dead easy to see. Next time you're going for a walk in the Yorkshire Dales, have a look at sheep and you'll be able to see their pupils rotate. I'll start to come to a conclusion now, so hopefully there's some time for some questions. I will talk briefly. Of all the work that I've done, this is the work that kind of ended up making the biggest noise in the press. And there was a kind of time of, the press kind of pick things up and then they kind of move on. But it was literally 48 hours in my life when all the newspapers were calling it up because they decided that this was an interesting piece of work to report on. And it was in many of the world's major newspapers, which was very nice. The reason I'm telling you this is because I thought it was very funny because different newspapers, of course, have different stereotypes. And the way they dealt with this completely fitted their stereotype. So the Daily Mail got hold of it and called me up and kind of got it wrong. And then it went online and then all their readers was going, this is outrageous. Why are we spending money on this? And I knew all this when I was at school at 5 years old, and this is a waste of money. So that was the Daily Mail readership. And then it went in the Guardian, and it turns out that the one animal that doesn't fit our hypothesis, don't know why, but one animal that doesn't is a mongoose. Look at a picture of a mongoose. They're very strange because they've got horizontal pupils, but they basically look like a small, furry, cute thing, and their eyes are facing forward, so they don't fit. So the Guardian readers were very excited because they go into a big debate. Is it mongoose or mongoose? This is This is what they were. They weren't at all worried about spending money on it. So it was, yeah, I just found it really funny. Okay, I shall come to a conclusion there, just to remind you back to my major conclusions at the beginning. You know, optics, really important in science. Optical instrumentation has been really important in the development of the science and still continues to be. And secondly, it's, you know, sometimes nice in life when we're working in, not just in science, but in academia in general, you can go down your own kind of very deep, narrow hole. And sometimes it's nice to kind of move between different areas and work with colleagues in different disciplines with the same time kind of keeping this general theme throughout. So thank you.
We've got some time for questions. I don't know if we've got anything in chat yet, but
let me just have a look. I think it's mostly about the
I should say this, the work in particular on animals was done in collaboration with a guy called Marty Banks, who's at Berkeley, and obviously colleagues of my former institution at Durham.
There's a short essay from Kirsten, who's had to dash off. So anyway, we'll attend to that. But I think that's it. So if anybody else would like to put a question, in the chat, you'd be very welcome.
Question over here.
You may have answered it. I was just going to ask which was the one ambush predator that had horrible flies. Is that
I think it's mongoose, yeah. Yeah.
You,
I don't know why.
why.
No, no idea why. Yeah, I mean, I guess, you know, things in nature don't work perfectly. Evolution doesn't give a perfect solution to things. Yeah. If go Google a mongoose, they do look weird.
Yeah, very nice talk. I was kind of wondering whether there's any opportunities for machine learning and AI for some of that image analysis. You know, you showed the effect of wavelength, and that kind of put me in mind that detecting as a function of wavelength and somehow pairing yourself up to see what it might look like.
Yeah.
Could you repeat the question?
Oh, so the question was, is there any opportunities for using machine learning or AI essentially when you're looking at blurred images to try and improve them? I guess that's the question. Yeah. Yes, is the short answer to that. And in fact, there's even before machine learning and AI became a popular thing, how do you take an image that's lousy and improve it? As a big area of research that's been going on since the 70s, and you've probably seen online, you know, the police take pictures of like a fuzzy number plate of some criminals they're kind of trying to catch, and they try and improve that. But there's an important distinction between the two. So if you're building a big telescope and you're trying to make a picture of something that's very faint up in the sky, okay, if you want to do machine learning or AI or whatever went before it, you have to have something there to measure. And the atmosphere is so awful that if you've got a faint signal, it's spread out so far away that you literally have no data that you can kind of work with. So if you've got tons of light, yes, machine learning and AI, definitely you can use. If you've got faint signals, which often you do in science, then you can't. There's also a subsidiary answer, a secondary answer, that often in science, taking pictures isn't the most important thing. You want to like measure the light, you want to send it off to a spectrograph or do spectroscopy, and you can't do that once you've recorded the light and done some clever tricks in machine learning. So you have to be able to correct the light in real time and then send it off somewhere else. So yes, you can do it, but there's kind of a bigger picture as well.
In the introduction, there was a reference to psychology as well, another level of this. Is there any evidence that people from different cultures see things in a different way, or better at seeing particular things?
That is definitely outside my sphere of expertise. I think that certainly different people from different backgrounds and races certainly more susceptible to the different eye diseases as they are, you know, disease in general. I don't think, as far as I know, that there's any evidence that they can somehow see better.
Just going back a bit, apparently American Indians
Mhm.
are particularly good at seeing angled shapes,
Ah,
rather
okay.
than uprights and horizontally.
Fascinating.
I don't know whether that is a common thing.
Yeah, I've not come across. And I guess then the question is that a culturally learned thing or is it a genetic thing?
Anything else on the chat, Rachel? Just check.
Go forward, mate. I guess that microscopy is really going to sort of lasers and small lasers and so on. So we're not using light optics to go below that 1 micron,
Yeah.
1,000 nanometer level. And if we want to see viruses, for example, we have to go currently to use an electron microscope, unless the virus is very big, which there are some, but not many. Do you have any thoughts about extending?
Yeah, I mean,
Can you just summarize what the
yeah.
was, otherwise
So
the chap people on Zoom won't pick it up, I'm afraid.
I guess the question was how might we extend, going down to greater resolution in microscopy. The great thing about microscopy is that you can get at your sample and you can do, it's not like the sky, there's nothing you can do, it's there, and or you can look. So there's a whole host of techniques in microscopy. For example, there's 2-photon microscopy. You can shoot a sample with 2 lasers and then it kind of excites it and you get light coming at a different wavelength, and you can use that to probe very fine details. So yeah, there's a whole range of techniques in microscopy.
Yeah.
But really, it's always, you can get this resolution limit of the wavelength of light. It's always a tough one to get around, and we are improving on that. But probably the highest ever resolution images are still down at X-ray.
So your image of the zebrafish,
That was with laser light. That was yeah. I mean, often that we're using the laser just because it's easier to kind of build stuff with rather than anything special about it from a sample perspective.
I guess the lasers have got cheaper and smaller.
More
powerful.
Simple lasers not expensive.
Or at the back.
A photograph showing the skyscraper. Surely that is something that we in the West would another culture.
I guess. Oh, sorry. The question was, I showed the picture of the skyscraper, and what effectively was that a cultural thing? We in the West are used to that. Maybe other cultures wouldn't. I mean, I guess I don't know the answer to your question without actually going to do a survey. But I still think people from wherever they are used to the concept. You don't have to show a skyscraper. You could show, a plant and bring the plant close to you. Still get that gradient in blur. I suspect that they will still see the same effect.
There's a question here on the screen. Perhaps you'd like to read it out and then a comment.
To the best of my recollection, BBC4 had a documentary which indicated that the Hubble Space Telescope was no longer going to be maintained. How will astronomy research continue if this occurs? Well, yes, the Hubble, I mean, I think the Hubble, first of all, is probably the most successful telescope ever to have been built. Well, you can argue whether Galileo's was more successful. Anyway, one of the most successful telescopes to have been built. It's been fantastic. It lived beyond its lifetime. I mean, I guess the simple answer is astronomy is still in a very good and healthy position. We've got the James Webb Space Telescope now, which is a larger, okay, it's got different instruments on it. But yeah, I think now is not a time to be pessimistic about astronomy. It's optimistic and a good time.
There will be a talk about the James Webb Telescope and more besides as the Priestley Lecture this year.
Are we done? In which case, I think it only remains for me to say thank you very much indeed, Gordon, for a fascinating tour of the world of optics. And certainly, I'm sure, certainly me, and I'm sure everybody else here will have learned something that they didn't know before. If only that a mongoose is an exception. we'll go away and look at mongoose with a different eye. But thank you very much indeed. That was really fascinating.
And it's mongooses apparently, not
Mongooses.
mongoose.
Mongooses. Mongooses with a different eye. Thank you very much.
Thank you.
And just to remind you that there are 3 more events that are already up there on the website, ready for booking. Bookings have been coming in. Do join us for the next events, and there will be more coming up shortly once we get all the details in place. So we've got on the 16th of April, Dr. Stephen Corns talking about Defoe's account of the populous and wealthy West Riding. It's 300 years exactly since Defoe came through Yorkshire, so we felt we had to give this one a whirl. And then Andrew Filarowski, recommended to us by our member of council, Christine Holstock here, and on Dies and Dying in Leeds, past and present. And that will be, Stephen Corns, that will be at the Leeds Library in Commercial Street. The next one, the Dies and Dying, will be here, we hope. And Professor Ian Brooks on inspirations from the atmosphere, his work not only as a scientist, but as a printmaker, which should be really fascinating. And that we hope will be here as well. So I hope you can join us. And those of you still out there, either online or in person. Thank you. The Leeds Library one, only in person. Should be marginally easy. So this is what we stop share. Yeah, and then.
Oh, right.